In the advanced field of genetic modeling, a significant puzzle emerged. Researchers developing complex deep learning models found that their sophisticated systems were being matched or even outperformed by simple statistical averages. This counterintuitive result presented a serious challenge to the prevailing assumptions about the power of AI in this domain. This slide illustrates the core of the problem. On one side, we have complex deep learning models, which were expected to provide highly accurate predictions of cellular responses. On the other, a simple statistical average or baseline. Yet recent reports indicated that the complex models failed to show a clear advantage. This wasn't a minor discrepancy. It raised fundamental questions about the utility of these advanced approaches. This observation was not anecdotal. As this quote from the source paper by Miller and colleagues indicates, these findings were being published in scientific literature. The reports were creating a growing consensus that perhaps these deep learning tools, despite their complexity, were not suited for this particular scientific task. This discrepancy led to a critical question. Was the issue with the models themselves? Were they incapable of capturing the underlying biology? Or was the problem located elsewhere, perhaps in the very methods used to measure their performance? This became the central question the researchers sought to answer. Here's the agenda for our explainer. We'll begin by examining the AI failure in more detail. Then consider the primary suspects, the models or the metrics. We'll look at the forensic framework developed to investigate the issue, see what it revealed, and observe the new picture that emerged. Finally, we'll discuss the broader importance of accurate measurement in science. So, let's first delve deeper into the context of this problem. To understand its significance, we need to understand the ultimate objective of this line of research. The primary goal here is ambitious, to create what are known as virtual cells. These are computational or in silico models that can predict a cell's response to specific genetic alterations. The practical application of such a technology would be to greatly accelerate the process of drug discovery by allowing researchers to screen potential therapeutic targets computationally. And this is where the roadblock appeared. The mean baseline is simply the average effect observed across all genetic perturbations in a dataset. It contains no information specific to any single gene. The fact that a sophisticated model designed to learn intricate and specific patterns could not outperform this blunt, non-specific average was deeply perplexing from a data science perspective. This paradox led to the investigation phase. Researchers considered two main possibilities. Either the AI models were indeed flawed, or the statistical metrics used to evaluate their performance were providing misleading results. This brings us to a key concept, metric calibration. A helpful analogy is a thermometer. If a thermometer is not properly calibrated, it cannot provide an accurate temperature reading, regardless of how well a heating system is working. Similarly, if a statistical metric is not well calibrated, it cannot reliably distinguish between a genuinely informative prediction and an uninformative one. So, the focus of the investigation shifted. Rather than attempting to develop even more complex models, the researchers turned their attention to the statistical rulers themselves. They began to operate under the hypothesis that the models might be performing adequately, but their performance was being incorrectly assessed. To test this hypothesis systematically, a new evaluation framework was needed, one designed specifically to assess the reliability of the metrics themselves. This framework involved several steps. First, following standard scientific practice, they established controls. For the negative control, a predictor that should perform poorly, they used the mean baseline. For the positive control, a predictor designed to perform well, they created a new method called an interpolated duplicate. This method generates a robustly informative prediction. Finally, they developed a new measure to quantify a metric's ability to tell these two controls apart. That new measure is the dynamic range fraction, or DRF. The DRF score quantifies a metric's calibration. In simple terms, it measures how well a metric can distinguish between a good prediction, our positive control, and a bad one, the negative control. A high DRF score indicates a well-calibrated, reliable metric, while a low score indicates an unreliable one. With this framework in place, the researchers could now systematically assess the commonly used metrics in the field. The results of this analysis were quite clear. This table summarizes their findings. Metrics that are common in machine learning, such as mean squared error, or MSE, were found to be poorly calibrated for this task, as shown by their low DRF scores. In contrast, metrics that were weighted or rank-based demonstrated good calibration. The evidence pointed not to the AI models, but to the uncalibrated metrics as the source of the problem. The underlying reason for this failure relates to the data itself. In genetics, the response to a perturbation often involves significant changes in only a small subset of genes. Poorly calibrated metrics like WMSE treat all genes equally, which means this sparse but important biological signal is effectively diluted by the noise from thousands of unaffected genes. It's analogous to trying to hear a single quiet voice in a large, noisy room. The next logical step, then, was to re-evaluate the performance of the AI models using only the well-calibrated metrics. When this was done, the outcome was substantially different. This chart shows model performance as measured by a poorly calibrated metric, WMSE. You can see that the scores for the deep learning model and the mean baseline are nearly identical. This is the result that led to the original confusion and the questioning of the model's utility. In contrast, this chart shows the same comparison using a well-calibrated metric, weighted WMSE. The difference is now unambiguous. The deep learning model clearly outperforms the mean baseline. The model's superior performance, which was previously obscured, is now visible. And this result was not an anomaly. The researchers confirmed this finding across numerous datasets and different modeling tasks. When the evaluation was performed correctly, the advanced models consistently showed a significant performance advantage over the simple baselines. The original contradiction was therefore resolved. The implications of this work extend beyond this specific area of AI research. It serves as an important reminder about the fundamentals of the scientific process. To summarize the key takeaways, first, what appears to be a failure in a model may in fact be a failure in its measurement. Second, this work helps resolve a major contradiction in the field, confirming that these AI models are effective tools. Third, it underscores that selecting an appropriate evaluation metric is as critical as designing the model itself. And finally, the path toward developing virtual cells for drug discovery is now on a more solid footing. This final quote from the paper encapsulates the central conclusion. The primary limitation was not the capability of the models, but rather the use of uncalibrated benchmarks that concealed their true performance. And this leaves us with a broader question to consider. If an issue of measurement could obscure progress in a highly quantitative field such as this one, it compels us to ask, in what other areas of science might apparent failures or limitations actually be artifacts of the way we choose to measure them?